Okay, well, thank you for waiting. We will get started with the webinar today. Today's session is about RNA-Seq analysis using Avadis NGS. The speaker is Ramesh Hariharan, the Chief Technology Officer of Strand, and today he will be demonstrating the key analysis endpoints that you can achieve using Avadis NGS. So if you have any questions throughout the presentation, please use the chat feature in WebEx, and we will respond to questions hopefully by the end of the session. After the session, we will be getting in touch with you directly to learn more about your interest in next-generation sequencing. So with that, we have Ramesh Hariharan.
Good morning or good afternoon, everyone, depending on which part of the world you are in. My name is Ramesh Hariharan, and I'm going to talk about the RNA-Seq features in Avadis NGS. Avadis NGS is our offering for analysis of NGS data. The crux of Avadis NGS and what differentiates it from other products is what is reflected in the tagline NGS for the rest of us. So Avadis NGS takes fairly large amounts of complex sequencing data and manages to bring all of that together in an interface that's very simple, easy to use for biologists, and is also very visually appealing. For those of you who are familiar with GeneSpring, GeneSpring is one of the leading expression analysis tools. Expression analysis tools that's been around for a while, and it's used by several hundreds, if not thousands of people. The interfaces and user experiences in Avadis NGS pretty much mimic the user interfaces in GeneSpring. So if you're familiar with GeneSpring, you will have no trouble at all getting started. Even if you're unfamiliar with GeneSpring, we believe that the interfaces are pretty nice and simple to get you started, and I'll show you glimpses as we go along. What Avadis NGS does is that it allows you to import aligned files and then run workflows for ChIP-seq, RNA-Seq, and DNA-Seq. So the thing to note is that Avadis NGS starts from the output of alignment. If you have reads obtained from your sequencer, then you need to put them through an alignment pipeline. It's very likely that wherever you got the sequencing done, the sequencing providers will probably already give you the service of aligning these reads against the genome of your choice, and will give you aligned reads in one of the formats listed here, SAM, BAM, or ELIND, Illumina ELIND text format. So you can take any of these files and import them directly into Avadis NGS. And you can run workflows for ChIP-seq, RNA-Seq, and DNA-Seq. Of course, the focus of the rest of this presentation is going to be on the RNA-Seq workflow. The questions that we'll seek to answer as regards to RNA-Seq using Avadis NGS are the following. If you have aligned data, how do you import it in? What all do you need to import in data? Once you've imported data, how do you determine the expression value for each gene from the reads that you've imported? Once you've determined the expression value for every gene, how do you normalize these values across samples? Once you've normalized expression values for each gene, how do you identify genes that are differentially expressed between experiment conditions? In addition to differential expression analysis, how do you determine differentially spliced genes, namely genes that whose overall expression may not be changing across conditions, but the mix of transcripts within for such genes would be changing across conditions. So if a gene has 3 transcripts, then let's say A, B, C, then the sum of the expression levels of A, B, and C may not change across conditions, but the relative proportion of A, B, and C, so A might be 50% in one condition and may go up to 80% in another condition, and likewise B may be 40% going down to 10%. And so that change in mix is what you're looking for when we do differentially spliced gene analysis. You can also look at variations. So how can you determine SNPs that are reflected in the transcriptome? And how can you determine fusion genes, so transcription products that are obtained from the combination of multiple genes? And finally, how do you determine new phenomena like discover new genes, new exons, new splice junctions? So this is the list of questions that we will address over the rest of this presentation. So to get started, the first question is, how is data imported in? To import in data, the very first thing you have to do is to specify the organism and the build that you need, that you're interested in working with. And you need all of the associated annotations, the gene and transcript annotations for this organism and build. One of the key points is that prepackaged annotations for a variety of organisms and builds have already been made available from the Avadis NGS website. So from the product itself, you can, through a couple of clicks, download prepackaged information comprising gene and transcript models, DB SNP IDs, reference sequences, and some more information for a number of organisms and builds of your interest. For instance, for humans, there's the HG18 and the HG19 builds. So for several organisms, there could be multiple builds. In case you're working with an organism or a build which is not available when you look for the annotations from Avadis NGS, feel free to get in touch with us. We've been quite good about responding to needs on demand as people request interest in working with specific organisms or specific builds. So once you've chosen the organism, the build, and the transcript model of your interest, note that there are 3 different transcript models that we have for humans and for several other organisms. There's the ensemble transcripts, there are REF-seq transcripts, and there are UCSC transcripts. So once you've chosen that, you can choose the platform. We support all the 3 most popular platforms you might be using for RNA-Seq: Illumina, the solid technology from Life Technologies, and 454 Roche. And then you need to choose specify whether your library was designed as a single-end fragment library, or was it a paired-end fragment library, or was it a mate-paired library. All the support for all these 3 common library types. So once you specify this information, you're ready to go to the next page where you specify the files, the input files of interest. Remember that Avadis NGS imports in pre-aligned files in SAM, BAM, and ELAND formats. So you can specify multiple files, and in the event that one of your samples is actually split over multiple files, you can rename, you can actually provide alternative alias names for each sample. And if one of your samples is split over multiple files, just give the same alias to both those files, and they'll get combined into a single sample. In this presentation, what you see on your slide is 3 replicates of a melanoma cell line, the MEWO, and 3 replicates of a leukemia cell, 2 replicates of a leukemia cell line. So with that, you're ready to import the data, and once you import it in, this is what the first look would be like. Let me go over the essential user experience elements in Avadis NGS. So work in Avadis NGS is organized into projects and experiments. A project comprises many experiments. These experiments could be a variety of types. One experiment could be RNA-Seq, one could be DNA-Seq. You could put them all into the same project, and potentially you could drag in information from multiple of these experiments into the same genome browser so that you can juxtapose information from DNA-Seq, RNA-Seq, and ChIP-Seq experiments. An experiment itself comprises a collection of samples that you're analyzing with respect to a particular biological question. And the analysis is driven by what we call workflows on the right. These are application-specific workflows, and they change depending upon whether the experiment that you have in focus is an RNA-Seq experiment or a DNA-Seq experiment or a ChIP-Seq experiment. And on the left out here, you have audit trails of all of your results. You start with a list of reads, which is called the all-aligned reads list. And as you perform your analysis, you'll create more read lists by potentially filtering this master read list. You'll create region lists, which are collections of regions on the genome. For instance, if you do SNP analysis, it'll create a new region list comprising the SNPs. You'll create new gene lists. If you find differentially expressed genes, that will create a list of genes. If you do pathway analysis, that will create pathways. If you do hierarchical clustering, that will create cluster trees and so on. All of these objects are stored in this hierarchy here, and they are stored in the order of in the audit trail order, so to say. So if you run an analysis on a particular list and create a new object as a result of that analysis, that object becomes the child of the parent object. And sitting right here in the center is the centerpiece, the genome browser, your window into the data. And we believe it's a fairly powerful and flexible genome browser that allows you to view a lot of the phenomena that we'll be talking about. Pretty much any of the objects that's listed in the audit trail list on the left, you can drag into the genome browser and view. Okay. So moving on to the next slide. So you've set up your import, you've imported in your data, you're familiar with the UI elements in Avadis NGS. Now you run through the workflows. The workflow on the right has a number of sections. One of the first key sections is the QC section, where we allow you to look at your reads and identify aberrant phenomena there. Here is a heat map where you can see that there's one row which clearly looks different from the other rows, and I'll interpret what this means. But essentially this reflects a bad tile where the base qualities of the reads in this tile are poor. And so you might, and there's a simple filtering function which would say, just remove all the reads in this tile. So to interpret this picture, this heat map out here, going to the right, you're essentially walking on a read, going from the beginning to the end of the read. As you go towards the end of the read, the quality typically decreases, and so the color changes from red to yellow. The color at the left, the beginning or the left end of the read, is typically red, which means the quality is good. However, on some tiles, so the rows correspond to tiles. On some tiles, it so happens that a lot of reads are bad even right at the beginning and not just while going towards the end. And that is reflected in an aberrant color all through the row. And that allows you to identify bad tiles and remove those. Likewise, on the left out here, you can identify tiles in which the number of reads which match exactly is dips for some reason. So the pink dots here, so on the x-axis in this plot you've got tiles. The y-axis represents numbers, so a pink dot indicates the number of reads in that tile that match exactly without any modifications, insertions, deletions to the reference. So for instance, this tile out here has about 35,000 reads that match exactly. You can see that the tiles on the left have a lot more reads that match exactly than the tiles on the right, so there's some strange phenomena going on there. And in particular, you can see out here there's a tile where the pink dips substantially, so the number of exact match reads dips substantially, while the red rises from below out here, which means that the number of mismatching reads rises substantially, while the number of exact match reads diminishes substantially, and that indicates a particularly bad tile which you might want to remove. Likewise, you can see views on, all of these are screenshots from the product. In fact, everything that you'll see in this presentation is exactly taken from a screenshot from the product, no external screenshots generated. So what you see out here is the distribution of reads by what we call mate status. So if you're importing in paired reads, then the two reads in a pair could be at a distance that is expected. Those are called normal reads, or they could be further away than expected, which signifies an insertion. This is called mate far reads, or closer than expected, which signifies a deletion, and so on. So you can look at this picture and often spot alarming trends. If something went wrong in the pairing and the alignment, then you'll have a whole lot of more mates missing, the pink ones, where one mate is aligned but the other hasn't, and that tells you that something went wrong, for instance. Then on the right, you have what we call the distribution of alignment scores. So every read matches the aligner that you ran before you brought data into Avadis NGS. That aligner will output for every read an alignment score, meaning it'll indicate the number of mismatches and gaps that are used to align a particular read. The greater the number of mismatches and gaps, the lower the alignment score. A read that matches exactly will get an alignment score of 100, and as you introduce more and more gaps and mismatches, the alignment score will reduce. And if you see that there are reads which have very low alignment scores, you might want to filter those out, because you don't know whether they really have that many mismatches and gaps, or is it just an alignment artifact to force an alignment of those reads. So essentially, you've got a number of such views into the reads to check if the reads look good or not, and if you want to remove some reads that do not look good. So once you've done this, you're ready to launch into the core RNA-Seq procedure. And let's get to the next question then. How are expression values determined for each gene? In a nutshell, expression values are determined simply by counting the number of reads that align to that particular gene. If you have 100 reads that align to a particular gene, then you say the count for that gene is 100. There are some intricacies there. So number one, only reads that overlap exonic regions are taken into account while counting the contribution, while determining the read counts. So here, for instance, you see that there are a number of reads here which are not overlapping exonic regions, in which case these are not counted while determining the gene's count. One exception to that rule would be if you have the novel detection switch turned on. So there's a novel detection switch that you can turn on, in which case the algorithm will also try to identify novel exons or novel genes. It will do so by looking at regions of the genome that have a strong coverage and no overlapping gene annotation. So there's no exon present in this region, for instance, but there is a fair amount of reads out here, in which case it will demarcate that region as putative exon or gene. Then it will see if that region is within the boundaries of a gene, in which case it will add it as a new exon to that gene. Otherwise, it will call it a new gene. If it's added as a new exon, depending on where it's located, it might call it an extension to an existing exon, or it might call it a new exon. And if the switch is turned on, indeed, then and such a region is found and added to the gene's model, then the counts associated with that exon will also be taken into account while determining the count for the gene. And another intricacy is that of multiply mapping reads. Aligners will often output, for the same read, they'll output multiple positions because they're not quite sure which of those multiple positions does this read come from. Very often this would happen for reads that come from repeat regions. So your input SAM or BAM file will have, for a particular read, many potential mapping locations for that read. In such a case, we take fractional contributions for such reads. So if a read maps at 10 positions, we take, and one of those positions is on this particular gene, then the contribution of that read to this gene's count is 1/10. So that's how we determine the count for a particular gene. There is an additional intricacy that comes from having overlapping genes. So here you see there's a gene on the positive strand, and there's an overlapping gene on the negative strand. And the question would be, how do you handle these reads here? Do they come from the gene on the positive strand? Do they come from the gene on the negative strand? Depending on the nature of the experiment that you've run, it may or may not be possible to say which of those is the case. So for Illumina data, unless it's a directional protocol, these reads will contribute to both of those genes. If it's ABI life tech data which has a directional protocol where you can separate out genes coming from either strand, then the appropriate reads will contribute, a particular read will contribute to the appropriate gene and not to both genes. Okay, so now that we know how we determine the read count for a particular gene, let's ask the question, how do we now take these read counts and normalize them across samples? There are two methods for doing that, one which is currently implemented in Avadis NGS, and the other which is not natively implemented but is yet, you can still perform this from Avadis NGS via a phenomenon called Transcripts, which I'll describe in a minute. Let me go over the current native normalization first. So the current native normalization determines what is called the RPKM for each gene. In the process, it essentially ends up normalizing by the total read counts within each sample. You take the total count of all the aligned reads in a sample, and you say, I want to make this total count the same for each sample. I'll do so by taking each sample and then multiplying the count by a certain number so that the total count on each sample becomes the same. And I'll use the same number to multiply the counts for each of the individual genes that we determined on the previous two slides. In a nutshell, that's the RPKM measure. More technically, it stands for reads per kilobase of gene length per million reads in the sample. So there are two factors here. You are saying reads per million reads in the sample. That is normalizing for different numbers of reads in each sample. And then you have an additional divisor per kilobase of gene length. That length division is academic in a sense, because if you're comparing genes across samples, then you would be dividing by the same length across samples, so it doesn't really matter. But it's become sort of a standard to use this RPKM measure as a measure of expression of a gene. And so we do provide RPKM computations in the process normalizing by total counts. A common question that's asked is there a way to visualize and look at the RPKM values and look at them in plots, etc.? And absolutely, you can, Avadis NGS computes RPKM values. It can show you RPKM values in scatter plots, profile plots, box whisker plots, etc. So you've got the whole works in terms of visualization of these values. A second normalization that's been doing the rounds more recently, and it stems out of criticism on the first normalization, the following criticism on the first normalization, namely that a small number of genes with very high counts end up skewing the total count of a sample, and therefore a more robust method to normalize across samples would not be to just look at total count, but be to look at something which is akin to using the median instead of a mean, and that goes as follows. So you create a pseudo sample from your collection of samples. You do so by taking for every gene its mean across all the samples. You take the geometric mean in this instance. So you've got a pseudo sample where each gene is represented by the geometric mean across all the samples. You take that pseudo sample and you take for every sample the ratio of that sample with this pseudo sample. So you get a column of values where some values are above 1, whereas the sample has higher counts in the pseudo sample, and some values are below 1. You take the median ratio here, and that gives you your indicator of, that gives you the number you want to equalize across samples. So for every sample, you take its ratio with respect to the pseudo sample, take the median ratio, and equalize that median ratio across all the samples. I mentioned this phenomenon called R-scripts that you can use. So from Avadis NGS, there's a way to call R, routines in R. R, as you all probably know, is a very popular open platform where a lot of cutting-edge method development happens and new methods are released. And from Avadis, if you install R on the same machine as your Avadis NGS installation, then we have a mechanism by which from Avadis NGS you can call routines in R. And indeed, you can use a popular routine that is present in R to perform this normalization and perform differential expression analysis based on this normalization. I'll show you how to get details of this later. It's up on our website. So once you've normalized the data, you can, as I mentioned, view these RPKM values. Here is a scatter plot of the RPKM values between two samples. Okay, so now we've learned how to import the data, we've learned how to determine gene counts, we've learned how we will normalize these gene counts across samples, and now we've got a nice set of normalized gene values, and we're all set for statistical analysis to identify differentially expressed genes and differentially spliced genes. There are again two approaches to statistical tests that Avadis NGS provides. One assumes, backing up a little bit, there's the background distribution assumptions in reads data coming from sequencing is in a way fundamentally different from signal values that come from microarray analysis. In the microarray world, you end up using t-tests and ANOVAs which assume a normal distribution. In the reads world, you don't have an underlying normal distribution. You have the errors are distributed in a Poisson manner. And Avadis NGS uses a certain transformation called the square root transformation to obtain an approximate normal distribution, after which all of these tests, t-tests and ANOVAs, all of those become applicable. And these are available natively within Avadis NGS for you to run and determine differentially expressed genes. An alternative method is currently available in R as an R bundle, and it's called DE-Seq, and it's been getting increasing prominence over the last few months. And again, via the R-script methodology, you can run DE-Seq from within Avadis NGS. You don't need to export the data, learn R, run things in R. It's as simple as as a click in Avadis NGS. The details of how to do this are available on our website, and I'll flash a screenshot later in the presentation. So in a nutshell, you have two different methods that you can use to determine differentially expressed genes. And the dialog for differentially expressed genes, the results when they come out, you'll see what you've been used to seeing in the microarray world, p-values, corrected p-values, fold changes, volcano plots, fold changes versus p-value volcano plots. And in the audit trail navigator, you'll create new gene lists, and which you can then use for visualization and further downstream analysis like gene ontology analysis, pathway analysis, all of the biological interpretation steps which I will not talk about in this presentation, but which are all present in Avadis NGS. This is a screenshot of a gene list that's obtained by differential expression analysis and showing only upregulated genes that are upregulated in leukemia relative to the melanoma cell line. The next topic, now that we've got a list of differentially expressed genes, is the determination of differentially spliced genes. So let me introduce you to the problem setting here. So imagine you've got a gene, let's say the RAP27A gene here, and it's got 4 transcripts. And for the 5 samples that we imported, the 3 melanoma and the 2 leukemia samples, here is a screenshot from our genome browser, and you can see very clearly that you've got overlapping, there are clumps of reads overlapping exonic regions in this gene. And the problem at hand is to figure out, you've got a clump of genes out here. How do you know, a clump of reads out here. How do you know for these reads, how many of them come from the first transcript, how many come from the second, how many from the third, how many from the fourth? What clues do you have to disambiguate this information? Clearly, looking at this picture, it doesn't look like one can make this determination. So there's a couple of clues that allow you to make this what we call deconvolution. And let me take an example and illustrate how this gets done. There are 4 transcripts here, and let's call them A, B, C, and D from top to bottom. And there are 2 conditions here. One is the melanoma condition, one is the leukemia condition. And you've got 2 profiles, one for each of these conditions. The red one is melanoma, the blue one is leukemia. You see each of these profiles is actually a string of dots, and each dot is aligned, it lies just below one of the exonic regions. And that dot represents a measure of expression, the RPKM, let's say, of the corresponding exon. So this dot here corresponds to this exon, this dot out here corresponds to this exon, and so on. So you've got, for every exon, you've got the profile for each of the 2 conditions. Now focus on this particular dot that is aligned with this exon here. This exon is present only in transcript A, and therefore you can conclude that transcript A has low expression in both conditions. So one of the key things that gives you a hint is the fact that there are exons which are unique to transcripts. And likewise, you can conclude that transcript C has low expression, that transcript B. So if you focus on this dot here, this overhang portion, it represents this overhang portion that's present only in transcript B. And so this tells you that transcript B is low in leukemia, high in melanoma, so it's downregulated in leukemia. And likewise, this one here is present only in transcript D, and so transcript D is upregulated in leukemia. So using unique exons, you can do this deconvolution. But even if there are no unique exons, there are additional hints to do this deconvolution. So for instance, if you've got reads that are spliced across exons, so a read in RNA-Seq could straddle multiple exons because the gene product, the transcript itself, has introns spliced out, and therefore what is contiguous on the transcript need not be contiguous on the genome when you map it back. So you could have a read that has a part in this exon here and a part in this exon here. And now if you have such junctions that are unique to a particular gene, then immediately you know that that read comes from that gene. So you have a number of such clues that tell you, at least for a subset of the reads, which transcript do they come from. And then once you've seeded your deconvolution with those reads, you extend them to the other reads by splitting the other reads in the same proportion in some sense. Now what I've talked about is very qualitative and conceptual. The actual procedure is a much more statistical, quantitative procedure that does this deconvolution. This is the same results shown in a slightly different picture. Here you see the same 4 transcripts, but each of the profiles here now represents one of the transcripts. You see one transcript D is going down between the leukemia cell line and the melanoma cell line. The other transcript B is going up, and the other 2 transcripts are low down below, they have low expression. And then this is the red profile, which represents the profile of the whole gene. You can see that the gene is changing, but only a little bit. But if you just looked at the gene, you would not see the deeper picture that emerges when you look at the transcripts after deconvolution. One transcript is going down strongly, the other is going up strongly. The gene is sort of changing only a little bit. So that is what transcript deconvolution is about. And to do alternative splicing analysis, on the next slide, you'll see the conceptual description of the algorithm. You use this deconvolution procedure to compute the proportion of a gene's count that can be ascribed to a particular transcript. So if a gene has total count 100, read count 100, what fraction of these reads come from transcript 1, what come from transcript 2? And the way you do this deconvolution is by using the procedure that I conceptually outlined on the previous 2 slides. So once you have proportions for each transcript in each of the samples or conditions, you just need to compare these proportions across conditions. If the proportions in one condition are very different from those in the other, then you say that the gene is differentially spliced. For instance, if there are 2 transcripts A and B, and A has 70% contribution in one condition and only 20% in the other, then you say that there is heavy differential splicing happening between the two conditions for this gene. Now, once you run this procedure, it will create you a list of differentially spliced genes. You can look at these sorts of pictures that I've shown on the previous slides to verify the differential splicing. And once you've verified the differential splicing, you can do either further downstream analysis, or you can, if you wanted to find genes which are differentially spliced but not differentially expressed, you could use the Venn diagram functionality to intersect the two lists and find regions that are unique to each of the lists. So you take the differentially expressed list, the differentially spliced list, and just take the symmetric difference, the part that is unique to each list. That will give you a list of genes that are differentially expressed and not spliced, and vice versa. And you can find the intersection as well, of course. So now that you've got lists of differentially expressed genes, lists of differentially spliced genes, let's move focus to variants. How do you determine variants in the transcriptome? In particular, SNPs, small insertions and deletions in the transcriptome. So Avadis NGS has a built-in SNP calling algorithm that's based on the MAQ, the MAQ algorithm, and this is the input dialog for that. It allows you to specify a minimum certificate that you need in terms of number of reads overlapping a particular position. And allows you to set filters for statistical significance. And once you set this, it runs through and it will give you a list of genes, a list of SNPs. You can drag that list of SNPs into the genome browser and visualize those, and this is how that view would look like. In the genome browser, you'll see a color-coded indicator, which is all the same color if it's a homozygous SNP, and if it's heterozygous, you'll see two colors, and you'll see the relative proportions in those colors. And you'll see the actual reads with the aberrant base mentioned below. You'll see the Avadis NGS SNP call. You'll see the DB SNP ID if there is if the SNP is indeed known in DB SNP. If not, then there won't be a DB SNP object in this DB SNP track here. And you can also drag in a repeat regions track that allows you to see if for every one of the SNPs that you have, is it in a repeat region or not, and you can filter those SNPs that are not in repeat regions very easily. Once you've determined SNPs of your interest, you can do further filtering on these SNPs based on their impact on the underlying transcript. Every SNP, if it's an exonic SNP, it could change the nature of the protein that's created because either it's a non-synonymous SNP and the amino acid that it's contained in changes, or if it causes a frame shift, or it causes a stop exon to be generated, or a stop codon to be generated, or a stop or a start codon to be lost. A number of such effects are possible, and Avadis NGS gives you the list of effects that Avadis NGS pronounces are listed out here in this list here. It also tells you which, if there's a non-synonymous change, then which is the amino acid change that happens. And you can filter SNPs based on any of these effects pretty easily by just checking or unchecking these boxes and then creating a list of SNPs of your interest. So that's how you can determine variants. Next, there are further variants which are not SNPs or small insertions, deletions, but whole combinations where chimeric transcripts are obtained from two different genes that get fused. And the question is, how does Avadis NGS determine such fusion genes? Here is an example in Avadis NGS it's a screenshot again of the genome browser where we've illustrated the famous BCR-ABL gene fusion that happens in leukemia cell lines. Here in the genome browser, there are lots of reads, but we've set filters on the genome browser to only see those reads which have their mates on another chromosome. So Avadis NGS, the genome browser, is fairly powerful. It has filters that you can set to just show you subsets of reads if you need to see only subsets of reads. In this case, you're looking at paired reads where one read of the pair is mapping here in chromosome 22 to the BCR gene, and the other read in the pair is translocated, meaning it's mapping on some other chromosome. And it so turns out that most of those mappings happen on the ABL gene on chromosome 9, which is out here. So the way you determine fusion genes is to see if there's sufficiently many paired reads that have one read of the pair in one gene and the other read of the pair in the other gene. And then if you have sufficient amount of such evidence, then you pronounce a gene fusion between these two genes. You also designate some of these gene fusions as read-through transcripts if they are adjacent genes where there are many pairs. So pairs are indicated by dotted lines. So two reads which are paired are connected by dotted lines in our genome browser, and if they are spliced, meaning that one part of the read maps to one exon and the other maps to another exon potentially on a different gene, then they are connected by a solid line. So if you have sufficient amount of paired and spliced read spanning evidence across two neighboring genes, then you call these as read-through transcripts. The very last topic is how are novel genes, novel exons, and spliced junctions detected. In a nutshell, the algorithm is simply to look for clumps of reads in regions where there are no genomic, genic, or exonic annotations. So here, for instance, this comes from a mouse myoblast time course study. For those of you who are familiar with the cufflinks procedure, this is the cufflinks, the data set that was used in the cufflinks paper. You can see there's a clump of reads here and there's no overlapping exon. There's only one transcript for this gene FHL3 and there's no exon overlapping here. So we call a new exon here, and this exon is added to this gene's model. So presumably there's a transcript that contains this exon. We of course don't determine what that transcript is, but we do add this exon to the gene's model and it contributes to the gene's count. If such a region was not present within the boundaries of a gene, then we would actually call a new gene instead of calling it a new exon within a known gene. Okay. The last novel discovery that Avadis NGS can help you make is those of novel splice junctions. As I mentioned earlier, a read that aligns split across two different exons is connected by a solid line in the Avadis NGS genome browser. You can call new splice junctions by looking for reads that span, that are connected, that are spliced across exons, across two exons which are not consecutive. So here you have the RNA-Seq transcripts, and you can see that there are a number of reads that are spliced from here to here, meaning they go from this exon to this one, skipping the one in the middle. That indicates that there is a transcript that has this exon and this exon contiguous, and this exon is skipped in that transcript. No such transcript is indicated in the RNA-Seq transcripts database, and therefore it looks like this is a new splice junction and a new transcript that one is determined. And indeed, in this particular case, we actually verified with the UCSC transcript that such a transcript was known. That transcript is the third transcript out here, the third one here in the UCSC transcript. So in the third transcript, the first and the third exons are together and the middle one is skipped. So that's how Avadis NGS determines new splice junctions. So that brings me to the end of the presentation. I'd want to leave you with a couple of pieces of information. One, you can go to www.avadisngs.com and download an evaluation version. When you try to download an evaluation version, the system will offer you a Mac, a Windows, or a Linux version, whichever you please to use. Minimum requirements for these versions are present here so that you can check those. All of these versions are intended to run on standard laptops as well. For instance, the machine I'm demonstrating from is a standard Mac laptop. Once you download and install a trial version, if you want to learn with the help of some demo datasets are available on the Avadis NGS website. In addition, there's a number of resources that we make available. The annotations for very many organisms and associated builds I've already talked about. And to reiterate, if the organism of your interest is not present on the website, then please feel free to get in touch with us, and we'll help create that package for you. In addition, you've got pathway databases present here, and you've got what we call a scripts library, which is our way of deploying additional functionality, delivering additional functionality to your Avadis NGS installation. So we send you a little text file with a few lines of programming. You put that, you open that in the Avadis NGS script editor. So it's from the menus. There's a particular menu that allows you to open that script, and once you open and run it, you'll get the functionality that you need, even though it was not present inside the product itself. There are two types of scripts. There are native scripts, and there are what we call R scripts. And I mentioned the DE-Seq methodology for differential expression analysis earlier, and this is where you can get the R script for that and instructions on how to run this R script. With that, we come to the end of this presentation. We'll be happy to take any questions that you might have.
Okay, thank you, Ramesh, for an informative presentation. That is the end of our webinar today.